Hello students, I am Professor Dr. Adarsh Kumar. Today I bring you the next learning episode in BSc Forensic Science of an important unit of paper forensic medicine, that is forensic entomology. As of now, we have discussed how to estimate time since death by observing various changes in the dead body in two learning episodes titled postmortem interval. Let us now learn the role of insects in estimating time since death, as well as other informations which can be applied in forensic context, that is forensic entomology. You will also be acquainted with entomotoxicology and tools and equipment required for evidence collection at the scene of crime. Dear students, I hope that the objective behind this chapter is clear to you all now. Let us now look the modules which we will be covering today. Module 1 will deal with the introduction to forensic entomology. Module 2 will deal with the current perspectives of forensic entomology. Module 3 will deal with the estimation of postmortem interval with entomology. Module 4 will cover entomotoxicology. In module 5 we will learn how to collect the evidences, the practical guidelines, and the module 6 will be the concluding modules. Introduction to forensic entomology. Before knowing what forensic entomology is, let me tell a quote on insects which says, quote, "The insects will tell you everything, people lie, but insects do not lie." Unquote. Forensic entomology is the use of insects and other arthropods that feed on decaying remains to aid legal investigation. Let us see the historical perspective of forensic entomology, how it developed. The earliest known reference about forensic entomology dates back to 13th century in a Chinese manuscript, The Washing Away of Wrongs, written by Sun Tzu. The first application of forensic entomology in determination of postmortem interval in a child death in France by Dr. Birgit de Herbois was done in 1850. Jean-Pierre Magnin, a French veterinarian, did revolutionary work to give the theory of predictable waves or successions of insects onto corpses. Bernard Greenberg is regarded as the father of modern forensic entomology. Now, let us see the divisions of forensic entomology. It can be urban, that is, which concerns with the litigations arising from bedbugs and termites affecting man-made structures and other aspects of human environment. Then there can be stored product, which covers the litigations arising from grains and other food contamination by insects. And third, where we are concerned, is the medico legal, which involves the analysis of necrophagous insects to gain insight into time since death, basically postmortem interval. So, what could be the role of forensic entomologist in this whole plethora? Identification of insects at various stages of their life cycle, then collection and preservation of insects as evidence, and then determining an estimate for the postmortem interval, that is the time between death and the discovery of the body, using factors such as insect evidence, weather condition, etc. And then testifying in the court to explain insect related evidence found at a crime scene. Now, let us see the forensically relevant insects. Insects are ubiquitous in nature. Even if we do not see them, they are likely to be involved in crime scene also. Entomologists can help forensic pathologists in determining postmortem interval by looking at the insects that are feeding on and around the body. Say flies. Flies are the first one to get attracted towards the dead bodies. These carrion flies include Calliphoridae, that is blowflies, then Sacrophagidae, that is flesh flies, and Muscidae, that is house flies, and these belong to order Diptera. These are the most widespread and accurate insects for determining postmortem interval. Then there are beetles. These are often found on old cadavers or in dry conditions. These beetles include Sylphidae group, that is carrion beetles, Astyphylinidae group, that is roe beetles, and dermistids, that is carpet beetles, and these belong to order Coleoptera. Then there are ants, third category. Ants, these generally consume smaller cadavers and belong to order Hymenoptera. All these insects belong to the class Insecta of the phylum Arthropoda. Then another perspective is the time of death, which with the help of larval development. The first approach in estimation of time since death is the estimation of maggots developing in the body. For this purpose, the knowledge of life histories of flies of families Calliphoridae, that is blowflies, Sacrophyridae, that is flesh flies, and Muscidae, that is house flies, has an important application in forensic medicine. The fly life cycle can take anywhere from few days to several months, depending upon several factors. For many fly species, the precise timing of their life cycle has been calculated and can be used to calibrate time of death or at least time of infestation. This blowfly metamorphosis, we should understand what is that. Blowflies are the gold standard forensic indicators. These are most useful in estimating postmortem interval. These are the ones that come in first immediately after the body is dead and start to decompose. They have an incredible sense of smell. They have a complete life cycle which consists of egg, larva, pupa and adult stages known as this complete metamorphosis. Let us see the blowfly life cycle. The first stage is the adult flies lays eggs on the carcass, especially at wound areas or around the openings in the body such as nose, eyes, ears and anus etc. Then second stage is these eggs hatch into larva, that is called as maggots, in around 12 to 24 hours. Then in third stage, this larva continues to grow by feeding on carps and molt, and then they shed their exoskeletons as they pass through the various instar stages. These are the first instar stage, that is around 5 millimeter long after about 1.8 days. Then second instar stage, that is 10 millimeter long after about 2.5 days. Then there is third instar stage, that is 14 to 16 millimeter long, that occurs after 4 to 5 days. Then fourth instar stage, the larva of length around 17 millimeter, and once it gets satiated, this develops into pupa after burrowing in the surrounding soil. And then in fifth stage, these adult flies emerge from pupa cases after 6 to 8 days. Insects have hard exoskeletons, so when they grow, they have to shed their skin. Larvae are all about growing and getting bigger, so they grow as fast as they can and molt when they need to, when their skin gets too tight. Once a larva reaches full size, it goes through metamorphosis, a complicated rearrangement of body parts and tissues that leads to the development of adult characteristics. Therefore, insects have so many stages in their life cycles. Now, let us understand the factors that affect the growth rate of insects. These are temperature. Higher the temperature, faster the insect will grow and develop into an adult. Then the food quality. Eating rich nutritious food help larvae grow faster. Then oxygen levels. Increasing oxygen concentration increases growth rate of insects. Then day length or the season. Many insects coordinate their developmental cycles with the seasons. And now let us understand the time since death after understanding time of death. Here we will understand the faunal succession. Faunal succession is the term used to describe the transition of colonizing species from one insect species to another through the different stages of decay. Succession is a less precise science than PMI, relying to some extent on the forensic entomologist's prior knowledge of a species over a wide range of families, and is also reliant on knowledge of local fauna to assess the significance of the presence or absence of a particular species. The faunal succession can vary significantly between individual cadavers, and the arrival times of certain species are influenced by local factors. Since insects invade bodies in waves, estimation of time since death requires the ability to identify each species in all stages of their life cycles and knowledge of the time occupied by each life stage under various conditions. Now, let us see the typical succession of insect colonization. These flies arrive within minutes or hours of death as the body begins to give off the odors of decomposition. The carrion beetles arrive within few days and dig underneath the body and begin feeding. The carpet beetles arrive once the corpse is dried out sufficiently and begin to consume skin and hair. Let us see the factors which affect the insect colonization. First is the weather. When it is raining, insects are less active, so colonization is slowed. Then the temperature. During cold, when temperature is less, insects are less active and grow slowly. Then the burial or exposure. Even a partially buried corpse decomposes slowly. Degree of exposure also affects how accessible the body is for colonization. Then location. Bodies in dry environment will desiccate before insect colonization. Bodies in wet places will attract a different set of insects like aquatic beetles. Then temperature dependent development. Insects are cold blooded organisms. Rate of development is more or less dependent on the ambient temperature. For each species there is a threshold temperature below which no development occurs. As temperature rises above this threshold, a certain amount of time is required for the insect to pass through each life stage. Because this heat is accumulated as thermal units, it can be calibrated and described as degree days or degree hours. Now, let us see the problems with the faunal succession. The differences in the weather, location, season and the extent of interference by other animals makes it very difficult to predict by a forensic entomologist what species will arrive when. As we know, the postmortem interval is the time elapsed since estimation of accurate postmortem interval, that is PMI, can help to identify both the criminal and victim by eliminating suspects. The estimated age of an immature insect that has fed on a body provides a minimum PMI because offsprings are not deposited on a live host. Basic assumptions that a forensic entomologist make before calculating PMI are: number 1, most homicides occur at night under cover of darkness when flies are presumably inactive. Flies will begin ovipositing as soon as they discover the body. Then faunal succession in and under the corpse will follow a predictable pattern. Then weather conditions recorded at a site distance from the death scene reflect the conditions at the scene. Then ambient air temperatures are the major factors influencing the rate of maggot development. Now, estimating postmortem interval, how it is done. Collect and preserve a sample prior to use in estimating PMI. Rear others to adult stage for identification. The moment of preservation is the point in time from which you calculate backwards to death, and use known developmental times at constant temperatures to estimate PMI. Then place of death. Since some of the species appear in a particular geographical area or a particular locality, from the examination of the insects, the particular geographic and ecologic place can be known. Then determine neglect of patient or children. Here also it is helpful. Cases where wounds and bed sores arising out of physical abuse or neglect become infested with insects, forensic entomology may be useful. Then also in manner of death. Actual event that led to death is especially important in advanced putrefied bodies. If putrefied bodies have wounds infested with insects, it could be homicide or trauma. In cases of death due to suspected poisoning, by using arthropods in a corpse or at a crime scene, investigators can determine whether toxins were present in a body at the time of death. Recovery of heavy metals from bodies in the 1970s and 80s led to the development of forensic research at the boundary between entomology and chemistry, and this became known as entomotoxicology. Entomotoxicology is the analysis of toxins in arthropods. mainly flies and beetles, that feed on carrion. This technique is a major advancement in forensics. Previously, such determinations were impossible in the case of severely decomposed bodies devoid of intoxicated tissue and bodily fluids. Entomotoxicology can be divided into two main categories. One, that is entomological analysis of substance induced changes to insect development rates with subsequent effects on PMI estimation, and 2, the toxicological analysis of drugs within insects. Substances may be more easily detected because of storage, excretion and bioaccumulation in insect metabolism, resulting in concentrations of substances higher than in the surrounding tissues. Substance induced changes can be caused by substances external to the body, for example, fire accelerants, oil, paint, organophosphate poisons, or those internal such as ingested or injected narcotics, alcohol consumption or pharmaceuticals. Most important in this context is the presence of pharmaceutical or narcotic drugs in the deceased and the differential effect these can have on insect growth. Depending on where in the body these drugs and their metabolites concentrate and whether some or all of the maggots have fed on those tissues. We know, for example, that over the counter medicines such as paracetamol is one of the commonest drug used in suicide, typically resulting in a level of 250 milligram per kg drug to body weight concentration. At this concentration, growth period of blowfly larvae is differentially increased by up to 4 days. This critical piece of evidence suggests that the growth pattern diverges from the normal linear relationship we more commonly use. A commonly prescribed drug for anxiety and sleep disorders, that is nordazepam and oxazepam, its metabolite in the human body, cause an increase of at least 24 hours, especially on or around day 4 of drug accumulation. The amitriptyline, a tricyclic antidepressant, prolongs the post feeding and pupa stages, resulting in death during either, thus preventing complete development to child adulthood. Another prescription drug, zopiclone, for the short term treatment of insomnia, which is very frequently used. This prevents pupariation, the process by which blowfly maggots turn into pupa. Acting on the peripheral nervous system, it has the effect of seriously disrupting the pupation process. Among the narcotics, cocaine seems to have little effect, but ecstasy, that is MDMA, that is 3,4 methylene dioxymethamphetamine, speeds up development and reduces both the larval and pupal growth periods, sometimes resulting in insect death, while methamphetamine on its own appears to have little effect other than possibly terminating insect growth at the pupa. On the other hand, human lethal dose of morphine results in a 24-hour increase in development, while heroin at human lethal doses increases the larval growth period and can dramatically alter the pupa duration by 18 to 36 hours. Organophosphate poisons such as malathion do not necessarily result in death of the flies breeding in bodies containing a human lethal dose. Instead, malathion extends the larval growth period by about 72 hours. Clearly, such alterations of the growth in different stages of the insect life cycle impact on the assessment of PMI. It therefore becomes critical that the entomologist is informed of the drug or alcohol uses in cases requiring PMI assessment. In cases where the body is extensively decomposed and tissues are no longer available to analyze, it may be possible to process the gut contents from particular developmental stages of insects collected from the death scene that have fed on the body. This may be the only recoverable evidence of drug presence in the decedent. Insects can be collected from a corpse or its surrounding in 3 groups, namely maggots, insects from soil and other insects. We will see one by one. Maggots are immersed and killed in very hot, almost boiling water and transferred to a solution of acetic alcohol, that is 3 parts, 70% alcohol and 1 part glacial acetic acid. Killing in hot water over immediate immersion in preservative has two advantages. One, death of maggot is instantaneous, so exact moment of death can be known, and second, hot water kills maggots in a relaxed stage in which they are fully stretched or extended. So, measurements of maggots can then be compared with measurements from the experimental work. If maggots are immersed in preservative to cool them, they will shrink and any measurement will be difficult to interpret. So, rearing of maggots can be done, that is they can be kept alive on some meat or liver and reared to adult stage. Any puparia either empty or with the pupa inside them should be preserved in a specimen tube. All the containers carrying live specimen must be perforated for gaseous exchange, so that live organisms get the proper oxygen. Collect specimens from everywhere on the corpse. Try to collect specimens of every type, shape and size. Sample size will vary depending on the number of larvae found. From all the larvae where fewer than 100 are available to 1 to 10% of the larvae where thousands are available. Then soil insects. The soil sample should be collected in specimen bags from which insects can later be extracted in the laboratory. Each soil sample should be as much as would fit into a pint bottle. Soil should not be compacted. Storage in ethanol ensures that a later DNA analysis for identification is still possible. This may be necessary if a morphological identification is not possible. Then let us see what are the equipments and tools required for evidence collection at the crime scene. These are fine paintbrush for collecting eggs, then spoons for collecting maggots, fine and medium forceps for collecting adults and the more fragile immature insects, then hand net for catching flying insects, ethanol 70% for storing dead specimens, then protocol sheets for writing down what specimens were collected, labels, then vials and storage boxes of different sizes for preserving living and dead insects, then sawdust or tissue paper for handling eggs and living larvae in vials or storage boxes, then shovel and robust plastic bags for soil samples and leaf litter, thermometer for measuring the body and ambient temperatures as well as the larval mass temperature, and then camera for the photographic evidence. Let us see the common sites for sampling from where it should be done. The natural orifices and eyes, then the traumatic wounds, at the corpse, the substrate interface and under the body, in the folds of cloths and pockets, shoes, socks, etc., from the carpet bag or material in which the body might have been wrapped, from the plastic body bag in which the corpse or the remains have been enclosed for transport to the place of autopsy and storage. To conclude this module on forensic and entomology, the use of insects in criminal investigation is well established. Usually the knowledge and experience of a professional entomologist is required to establish species identity and to understand and explain the biology associated with those species. The most frequent application of insects to criminal investigation is the estimation of a post-mortem interval, that is the minimum period since the first eggs were laid. Other important applications of forensic entomology include the assessment of drug use, that is entomotoxicology. In addition to no post-mortem interval, knowledge of forensic entomology can also be applied to know the place of death, determine neglect of patients or children as well as the manner of death in some cases. The blowflies are the gold standard forensic indicators, especially in estimating PMI. These are the ones that come in first immediately after the body is dead and start to decompose. They have incredible sense of smell, they have a complete life cycle which consists of egg, larva, pupa and adult stages. Then the equipment and tools for evidence collection include fine paintbrush for collecting eggs, spoons for collecting maggots, fine and medium forceps for collecting adults and the more fragile immature insects, hand net for catching flying insects, ethanol 70%, protocol sheets, labels, vials and storage boxes, sawdust or tissue paper, shovel and robust plastic bags, thermometer and camera for photographic evidences. Sampling can be done from natural orifices and eyes, traumatic wounds under the body, in the folds of cloths and pockets, shoes, socks etc., from the carpet bag or material in which the body might have been wrapped and the plastic body bag in which the corpse or the remains have been enclosed for transport to the place of autopsy and storage. Dear students, with this module of conclusion, our chapter comes to an end. I hope you all have enjoyed this chapter and gained valuable information regarding forensic entomology and entomotoxicology. Do keep in mind what we discussed today. I will be back with one more lecture in this series. If you want to learn more and enhance your knowledge, you may log on to our website www.cec.nic.in for MCQs, quizzes, LORs etc. Make sure you revise the modules frequently so that you master the topic well and take up the exercises. Thank you for your time today. I will see you in the next lecture. Keep learning, work hard and do revise the chapter. Goodbye.